workshop expecting them to tell me how this organism can be used in genetics and development. But as it turns out, it has not been used much in genetics and development. So actually it was my job to come up and tell them how I would use this organism in genetics and development. So we worked for a couple days in trying to determine how this could be done, and Tracy and I, came up with an idea, and we're going to be presenting you guys with that idea today. So as far as what to expect, I don't know. We'll find out. We'll find out together. So let me just tell you what we're going to be working on and why. Oh, also I forgot to mention. So this is especially interesting to me, not only because I help with genetics, but the last part of the title of this course is Genetics and Development. And I'm an embryologist. I did all my work in embryology at UVA, so I love the embryo. So this is a good opportunity for me to kind of bring in my background and investigate some issue that has not been investigated for your class for the last couple of weeks. So we'll be doing it this week. You guys go home for a week, and then we come back and we're going to look at our results and try to figure out what worked, what could be improved upon, etc. And you guys are the starting of incorporating a new lab series into genetics and development here at JMU. So it's actually a pretty important process, and you're like the first ones to help develop it. So thank you for that. You are, in fact, my guinea pigs. So let me tell you what we're going to work on, first of all, and definitely if I'm going too fast, or if you need clarification, or you want me to repeat something, raise your hand and we'll stop and talk about whatever it is that you need to talk about. So the big question that we're going to be talking about in here, for our purposes, is the role of insecticide on the development of Calissobruchus maculatus. I call them the bean beetle. And so let me tell you a little bit about the bean beetle, first of all. They are beetles. I'm not a huge fan of bugs. They kind of freak me out. But as far as bugs go, these are actually pretty cute. They're kind of right up there with ladybug. They're very small. They crawl. They don't really fly around. They're tiny. They're not disgusting. And in fact, I think even more kind of creepy for me than beetles are the larvae, like the maggots. The maggots for these are like nicely held inside of a bean. So they're not like writhing masses of movement. And that freaks me out. Did you guys get freaked out by your flies? Yeah. So this is not quite so scary, and they're kind of cute. So we call them the bean beetle, mainly because they deposit their eggs on the beans, and the beans remains the nourishment source for the developing larvae until they eclose as adults, and then they walk around on the beans, they mate on the beans, and they lay eggs on the beans. So that's why they're called the bean beetle. When I was doing a literature search, I found that these were also referred to commonly as a cowpea weevil, because they often infect cowpeas, which we call black-eyed peas. So if you see on your desk, you've got a petri dish. The bottom petri dish, the largest one, contains black-eyed peas. And we're going to be specifically focusing on the black-eyed peas today. So some other interesting things about these guys, they are considered agricultural pests in very warm climates, particularly in Asia and Africa. They're not really a problem in North America because we have a winter period that gets too cold for them to be able to survive. So we still have to treat them as a pest. They're called a stored product pest because typically they will infest dried beans. So when we are finished with our experiments, I'll be putting them in the freezer for 3 days, and then we'll be throwing away those cultures. So hopefully that's not too scary for anybody. That's what we do to your flies. So one of the really, really, really cool things, and I appreciate it because, like I said, I don't really like looking at larvae. These larvae they are laid as an egg on the surface of a bean, and then they go inside the bean, and they eat and eat and eat the endosperm until it's time for them to pupate, and they emerge as a fully grown adult. So the bean serves as a reservoir, and if you think about it, it's kind of like being in a womb, because they're surrounded by this object, they're isolated from the rest of the world, and that's where all the growth and development takes place until it's time for them to emerge as an adult. So that's a really cool thing that we're going to be using to our benefit. Another really cool thing is like the flies, these are sexually dimorphic, which means that the males look different from the females. But the really good news is you don't need a microscope to tell the difference between the two, and we're not collecting virgins. So that's like, yay! That's really good news, but at the same time, it's really easy to tell the difference between males and females. And this is actually something that I was a little bit worried about because It took me a while to be able to tell the differences between male and female flies when I was learning that as an undergrad, and I thought, oh, not another one of these. These are super simple. I could tell the first time I compared the two, which one was male and which one was a female. So these are kind of off scale. This is a male, over here is a female. I tend to think that the males run a little bit smaller than the females, but the females have this really very clearly different posterior. So they have this extra little bit on the very posterior, and it's got pigmentation. It looks just like that. And it's really easy to see. You can just see it by looking with your eyes. And if you really want to see it, you can put it on your dissecting scopes and take a look at it that way as well. So one other thing to mention about this, this morph is that these beetles are actually sedentary when the beans are at a high enough density. So if there's a lot of beans in proportion to the beetles, they don't fly, they just walk around. So that's really good for us too, because we're not going to open up our dishes and have to deal with flying around beetles. That would be miserable. So they are sedentary. They may get a little scared. They do play dead very well, actually, when they get jostled around, but they are sedentary. So when you take the lid off, you'll kind of tap it like you do with your flies to keep them out down at the bottom, but they're not going to be flying out. So it's a little bit easier to work with. Everybody with me so far? Okay. So in terms of the life cycle, it's actually really similar to your Drosophila. One thing that's different is that they are sexually mature 24 to 36 hours, so you've got a little bit more time to work with if you're interested in collecting virgins. We're not going to do that for today. The males will seek out females to inseminate. I was looking at some cultures today, and there were some males in there, and the males were like mounting dead females, which was really weird. But that's what they do. So the males will seek the females to inseminate. The females will store that sperm in this structure called the spermatheca, and the spermatheca will allow for each mature egg to be fertilized as it is laid, and then that fertilized egg is laid on the bean. It stays there until the embryo is ready to emerge from the egg casing, and then the larva burrows into the seed coat, through the seed coat, into the seed, eats the endosperm, goes through a couple molts as the larva, so first instar larva, second, third, fourth instar larva. Now it's really big and fat. And then it goes into pupation, and it molts into a beautiful adult bean beetle. So that was a little bit more information than I had intended to give, but I'll go over that a little bit more slowly in just a second. One thing that I definitely want to mention, and it's kind of cool for what we're doing today, is that during their normal adulthood, the beetles don't eat. They may drink some water if it's presented. We're not going to present them with water because I don't want to drown anything, but they don't eat. As an adult, all they do is have sex and die. That's it. And so we're going to use that to our advantage. So let me just go over some of these things that I may not have mentioned. So once inseminated, adult females will oviposit or lay an egg on the surface of a bean. The larva hatches from that egg, goes into the bean, it burrows through the seed coat, and it goes into the endosperm. So let's say this is our bean. And I definitely encourage you guys to pick up some of the little dishes and take a look at the beans there. You can see the egg. They're really small, but you can still see them pretty easily. So what happens is this goes through some embryogenesis, and then the first instar larva hatches from that, and it eats or chews through the seed coat that surrounds the seed or bean, and it goes into the endosperm, and it's going to burrow. So it's got these really strong mouth parts and some enzymes that's going to digest the endosperm. So if you think about the inside of a bean that's dry, it's really hard. So there's some serious enzymatic activity going on with these larvae. So when it goes into the endosperm, it's going head first and chewing its way. So this is kind of an interesting thing. So as it's going in, it's also eating, so it's digesting, and so the fecal matter actually gets pushed out in the back, and it actually starts filling up the egg casing that it came from. So it's filled with frass or fecal material, and it kind of has this speckled or whitish color to it once it is completely filled up. Isn't that weird? It's like so weird. So anyway, this little guy goes in here. He goes through the first instar larva, second, third. Now he's a really big fat larva, like that book by Eric Carle, The Very Hungry Caterpillar. So now he's a very fat larva. And what's going to happen is it pupates, undergoes metamorphosis, and eventually it's going to come out of this being a fully formed adult. And I just think it's cool. Like, it's kind of neat. So let's see, I think I've got all of this. Right before the pupa comes out, if you take a look at the beans, you can see a window, and I have a picture of that, so I'll save it for you. I'm not going to try to draw it for you. But you can see like a window of the seed coat that's covering where the pupa is going to emerge out from, and then they just go and they find a mate, and they mate, and the process of life starts over again. So here are some really cool pictures. First thing I'll notice, or I'll tell you, is that these bean beetles have been raised on mung beans, which are the small green beans that you see in the tiny little petri dishes. Definitely take a look at those. You'll be able to see the bean beetles. You'll be able to see the beans on which they have been surviving. You'll be able to see eggs. You'll be able to see holes in the beans, and that's where an adult has emerged. You'll see all sorts of fun things in there, and that's going to be important when we take our observations when we come back after Thanksgiving. So the first picture on the top left is a mung bean with an egg being pointed out to you. In high density, you may see more than one egg on a bean, but one bean does not support more than a couple, one or two larvae at a time. So the rest will just die. So the next picture on the top is a picture of a mung bean that has been opened, and they're really hard to open. So some force was involved. And then the larva was taken out of that. They are kind of ugly, but they're not like slimy, and they don't like slosh, which our Drosophila does. So I think that as far as larvae goes, they're not cute, but they're not as ugly as the fruit flies. So that's what that looks like. Again, this is all happening inside the bean mill. So just before they emerge as an adult, you can look at the bean, and now we're on a black eye. You can look at the bean and see like the seed coat is the only thing in that area. Underneath is hollow. And the beetle is actually just beneath that. So his last little bit before he comes out is that he's going to chew through the seed coat and just climb right out. And that's all there is to it. So here in this photo is a picture of a brand new pupated beetle. So they have no pigmentation, but as time goes on, they develop the pigmentation. So the next picture is the beetle right before it's getting ready to eclose or come out of that bean. And then again, they lay their eggs on their beans. They emerge through the seed coat here, and then once they emerge, they leave behind this hole. And they're perfectly circular. They look like they've been drilled. That is indication to you that a bean beetle has been there. So now, since I have taken this course and I've learned about bean beetles, I'll notice these little circular things sometimes when I go to markets and look at beans, particularly organic, and the big bulk things, and I kind of am a little bit freaked out. So now you'll be freaked out too. So what's known about the genetics? Actually, fair amount. So we know that the chitin number for chromosomes for the bean beetles is 20. So they have 20 chromosomes, just like us, and just like your Drosophila. The males are the heterogametic, so they have an X and a Y, and the females have two X's. And that's what the karyotype looks like. In terms of genetics, about 80 genes have been identified in terms of what they do and expression patterns. The sequencing of the entire genome is actually currently underway, so there may be some work that comes out from this, but right now that analysis has not been completed. So in terms of development, we're kind of thinking about both of these things at the same time. So we've got genetic programming that helps to pattern the development of these animals. So what we know, fertilization occurs as females lay the eggs, glue them to the surface of the bean. Early embryonic development occurs inside the egg. Once the first instar larva is ready to come out of the egg casing, it burrows through the seed coat and into the bean. Development is actually pretty similar to Drosophila development. We know that. They have segments determined at the end of this period called the blastoderm formation, which is very similar to Drosophila. There are 4 larval instars, so they get progressively larger and larger and larger and larger, and then they pupate until they form an adult. So pupation occurs inside the seed, I can't even talk. Pupation occurs inside the seed, and then the adult emerges from that. And just like most things that develop temperature and humidity do have an effect on this. So the higher the temperature to a certain point, you have faster development. So you can actually speed up or slow down development by modulating the temperature. And the same is true for humidity. So the humidity in this building is actually pretty low. So I've had to incubate these in the presence of large containers of water. Otherwise, the bean beetles will actually die. So that's something to keep in mind as well. So there was a paper published in the 70s, and it's got a picture of what the larva looks like. And this should make you very excited, but you don't know what to be excited about, so let me tell you. It's very, very cool. So this looks a heck of a lot like the Drosophila development. In fact, Ms. Kaylin can tell you that the class yesterday prepared some cuticles, and they expect to see patterns that are similar to this. So what is this? This is a larva that has been cleared of all of its contents, and the only thing that you see is the outside cuticle. And on this cuticle, I can tell you what is anterior and posterior. I can tell you what's dorsal and ventral, and I can tell you how many segments are present. So because these creatures have to dig through and chew through a lot of really hard endosperm, they have pretty significant mouth parts, and they're darkly colored, they're black. And so I know that this is anterior, and the opposite tapered end is posterior. On the bottom surface, or ventral belly surface, you have these denticles, which are, I think, easier to see on the illustration. And these denticles sort of are like grippies on the bottom of your slipper socks, and they help the creature be able to navigate the inside surface of the bean. And you can see these structures pretty well under dark field illumination. So opposite that is the back, or the dorsal edge. So these cuticle preparations are really good because they tell us a lot about the development of the larva. And in addition, we know that there are 11 abdominal segments and 3 thoracic segments. Okay? So that's what we know. 11 plus 3. You're like, where is this going? Trust me, I promise I'm getting there. So where is this going? There has been a lot of talk recently about the role of insecticides in the environment. In fact, one of your bioethics questions was on GMOs, and it mentioned a specific topic in that it was in some GMOs there is the inclusion of a bacillus toxin gene that allows for larvae to be killed. So that's where this starts. We're going to be using BT toxin today. So my question is, what is the effect of the environment on the development of these organisms? So if they have wild-type parents, unless something really random happened to the genome, they should develop in a very similar way to their parents. And they should all develop in the same way, right? So what is the role of exposing these larvae to an insecticide? Is there going to be a change in the development? And if so, that's not due to their genetics, that's due to their exposure of something that changes the original genetic programming. So in humans, we call that exposure to a teratogen, or exposure to a mutagen, if you're specifically changing a genome sequence. So here I have one of my favorite images. Like I said, I'm an embryologist, and this is from the World Health Organization, but it basically depicts what goes on during human development after fertilization. So you can see that lots and lots of body structures are being patterned and formed during development. So the central nervous system starts being laid down at week 3 of conception. Most of the time, women don't even know they're pregnant during week 3, and it continues until the birth of the child. So similar developmental processes happen in our larvae. And what we're going to do is take a look at the morphology, or the body, to determine whether or not exposure to these insecticides is changing that normal programming. So be thinking about how we can do that and how we know what to gauge as normal. And then the little picture I have on the other side are just some common mutagens that we know causes a mutation in the DNA. So can anybody tell me what a teratogen is, like one that has historical notoriety? Have you talked about teratogens at all? Alcohol, yes. So what does alcohol cause? Fetal alcohol syndrome. So it causes a lot of neurological defects, actually facial deformations. The eyes get really close together and sort of at an angle. There's less learning in children born with fetal alcohol syndrome. That's definitely one. What else? Have you guys heard of thalidomide? Does anybody know what thalidomide was used for?
Morning sickness?
It was used for morning sickness, that's right. So I've been pregnant twice. I have 2 children. And I had morning sickness with my daughter morning, noon, and night for 40 weeks, and with my son, just thank God, the first trimester. But trust me, when you're like throwing up and all you think about is throwing up, you seek for any relief you can get. And at this point of time, in about the 1960s, not a lot of research was being done on the effects of drugs on unborn children. So these women, particularly in Germany, were getting this drug, which we call thalidomide. And thalidomide would help alleviate all those morning sickness problems. But then the babies were being born with this condition called phocomelia, and that is characterized by having flipper-like appendages. So like hands at the shoulders and feet very, very close to the hips. So all the segments, arm and bone segments, arms and legs are being completely diminished. It's not because their genes were messed up. It was because the presence of that drug was messing up how those genes were being interpreted. So my question is, does exposure to this toxin cause for some sort of morphological change in our larvae? So what we're going to use is the Bacillus thuringiensis toxin. It's also called BT toxin. This is a toxin that's actually made by bacteria, and it's been used since about 1920 by farmers in agriculture. It creates something called a delta endotoxin, and that endotoxin is crystallized until it's ingested. And by the processes of digestion within insects particularly, the endotoxin is cleaved to a very active form that starts to basically dissolve the digestive systems from the inside out. So these creatures that ingest the toxin will start eating less and less and then just die. And when they're dissected, you can see that the entire intestinal tract is just grossly deformed, and there are a lot of problems because of the toxin. So it's regarded as environmentally friendly. It is thought that there is not a lot of problems that come forth from using it on birds and mammals or humans, so it's considered environmentally friendly. And in fact, people use this in large amounts and still have crops that are considered organic. So that's one thing to keep in mind. So one thing, this is like the perfect pesticide because it supposedly doesn't have very many negative impacts on the environment, but we are seeing now that some organisms, particularly larvae of the organisms, are developing a resistance to this toxin. So that's definitely something worth noting. Another insecticide I'm going to talk about, and it's up to you whether you use it or not. I will say this, it has been linked with causing problems with humans. So some reactions, like skin reactions, it's thought to maybe induce ADHD in children. So I have it. If you use it, you'll have to use it with me standing right beside you and under a fume hood. If you don't want to use it and don't want to like stress about whether or not these are real effects, we'll just use BT toxin. In either case, we're going to use gloves. So malathion is an organophosphate. It targets processes so that it's a neurotoxin and it causes death. And it's an oil base, so it gets all over your organism and it kills the beetles pretty quickly, from what I saw. So what it's used for commonly, it was used to combat West Nile virus in New York City. So it was just spread over large areas. It's also used in treatments for head lice, body lice, and scabies. So it can be used on your skin, but it's at a very low concentration. And here's kind of why I mean, it's available, but I would hate for anybody to lose their memory the week before exam week. So it's up to you. I'm making this seem very, very appealing, I know. So our particular malathion is diluted 1 to 10 million. I had it at 1 to 1 million, but I went ahead and diluted it 10 times further just to be safe. My husband's brother-in-law runs a pesticide company, and he told me that this is a safe concentration, so 1 part per million is good. I went ahead and went one step further and did 1 part per 10 million. And it is available. I will stand right beside you. I've been working with it for a couple of weeks now, and I haven't lost my memory to my knowledge. So it's definitely available. Okay, so this is the last slide. So what I need you guys to do, I'm going to ask you to work in groups of 6 today, so 4 tables, and you guys are going to design an experiment. And here's our question. Does exposure to insecticide interfere with larval development? And I'm just going to go through a few of these, and then we're going to get into individual groups and start talking about this. So I'm going to skip over the first one because I want each group to kind of come up with their own experiment, but describe an experiment designed to address the question posed. And then predict the outcomes for each possible interaction. So what are the predictions that you would have? Okay, so a prediction could be exposure to insecticide is not going to cause any problems with larval development. Sure. Any other predictions? How about the opposite? Okay, so exposure to insecticide will cause a problem with larval development. Do you think that it's possible that we might not see some of the problems that happen with these larvae? So maybe it's going to cause a problem, but we can't see it. And that's actually something I hope to address next semester as well. We're working on a biochemical assay to test whether that's true. Identify and list variables you would manipulate in your experiment. So what are we going to be doing to these bean beetles? Yeah, we're going to expose them. And how are we going to expose them? There are plenty of different ways. So I had 4 groups do this yesterday, and they came up with 4 different ways. So you need to think about how are we going to expose these insects to insecticide. And remember, the adults don't eat it. That's a huge advantage, because the only way that it can kill them is if they ingest it. So think, what are your advantages to working with this system, and how are we going to target so that the larvae are exposed? Okay? So what are some of the things that we need to keep constant? Right. So exactly. So what kind of things affect the developmental status of our organisms?
Temperature.
Exactly. So pH, temperature, humidity. We have to keep that constant, because if I kept one in my office and one in the incubator, and I saw huge differences in the size of the larva, it's probably not because of the insecticide. It's probably because they're at different temperatures. So we've got to keep everything constant except for the exposure of pesticide. So list the data you would collect to determine if your predictions were true. How are we going to tell if the development is affected? So are we going to look at the beetles that emerge? Possibly. Something I forgot to tell you, I'll tell you now, it takes a couple weeks for them to emerge. So the reason why we're using the black-eyed peas is they're really easy to break open. So what are we going to do? We're going to break them open, and we're going to look at them. So you're going to look at the head, you're going to look at the posterior, you're going to look at the abdominal segments, you're going to look at the thoracic segments, and you're going to see if there are any gross defects between our control group that's being kept at a constant temperature and pH as compared with our experimentals, but not exposed to pesticide. Yes?
Seems like each different beetle would be laying eggs at different times, so they would be at like a different stage of life. Would that have an effect on what, like the mutation, I guess the hypothetical mutations would look like?
Sure.
If it was like a really small larva and a large one.
That's right. So what should we do? How could we prevent seeing larvae from different developmental stages?
Classify them based on size.
Sure, we could classify them. That's a great idea. Did you hear what she said? She said take the parents out after a certain period of time. That's a great idea. So maybe before you go home for break, is everybody leaving on Friday night? So I would say before you go home for break, just take the parents out, and we can get rid of those, and then we'll have larvae. Are you leaving like right after class today? Hopefully not. Someone in your group will be here. And they can take the parents out. I would say they probably need a day or 2. And if you have problems with that, I will actually be here until Saturday. So you can leave a note, and I can take the parents out for you. Okay? That sounds like a reasonable deal to me. All right. Describe data analysis necessary. We talked about that. We're going to look at changes. And we may be doing some measurements with a ruler, but we're not going to get much more serious than that for this point. And then graphical outcome. So if I were to make a graph, and I would say exposure, that's spelled wrong, to insecticide. So I have my wild type or control, and I have those exposed. How could I graphically present this?
Bar graph? A bar graph?
Bar graph. I think that's a great idea. So maybe different levels of exposure, that sort of thing, and measurement of the length of the larva. So just think about how you want to be, what characteristics you want to be actually measuring. Is everybody okay with this? All right, so on your desk you have some bean beetles. If you need more, just let me know. I've got plenty. But what I want you to do is look at them. Look at them, see if you can see male versus female. Look at the beans. Look what it looks like when they emerge from their beans. You have black-eyed peas, and you need to design an experiment, and you have anything in here to use. I have the Bt toxin up here. Please use gloves. I have paintbrushes, spray bottles, micropipetters. You just tell me what you need.
You can kind of tell the males from the female, but sometimes